everybody. This is our final set of notes for the semester. Next week is semester exams. I'll be going over that with you guys a little bit in the live session. So I'm going to jump right into the notes, get going, and then we will be getting ready for semester exams next week. We'll probably have a good review day next week, Tuesday, and then your exam is on Friday. So without further ado, let's go ahead and get going on the notes. So last lesson in Module 5, still talking about DNA, RNA, and proteins. Today we're going to focus on a specific type of protein known as an enzyme. So we're on Module 5, Lesson 3. There's your learning goals for the week, and it's also on Canvas, so you can check those out. And we're going to be talking about proteins and specifically this time around enzymes, okay? All right, so have you ever used hydrogen peroxide? So if you've ever gotten a cut or skinned your knee or gotten a cut on your finger or something like that, a lot of times what people will do is they'll pour hydrogen peroxide on it to basically kind of clean out the wound, and it kind of bubbles. Well, the bubbling is basically from an enzyme called peroxidase that's found in your cells. And peroxidase breaks down hydrogen peroxide into water and oxygen. So it's releasing these oxygen bubbles, which is where the bubbles are coming from. The reason that this is happening is it's doing it's basically breaking down. It's doing a chemical breakdown of hydrogen peroxide into those little parts. In fact, it converts 5 million molecules of hydrogen peroxide per second into water and oxygen. So it speeds up the reaction. Hydrogen peroxide would actually break down into those components naturally on its own, but it would take a long, long time without the peroxidase enzyme. So if you look at the image to the right, it's showing an enzyme, a peroxidase enzyme, which is basically a special kind of protein. This enzyme is found in most living things. In humans, we have peroxidase among other places in our blood, in other tissues as well. This is just one example of 100,000 or so different proteins that our body could make. Each protein has a specific task. In this lesson, we're going to look at what those roles are and how proteins kind of are essential for life and how enzymes kind of function to help keep everything kind of functioning in our body, right? So remember, DNA is the instructions on how to make proteins, which is why we're covering proteins right now, and then proteins do all of these metabolic activities. Proteins also control a lot of our physical characteristics. So if someone asks you to picture a cereal bowl in your mind with a specific shape or structure, kind of comes into view, right? The structure of the bowl lends to its function. It has to hold cereal, milk, and allow the user the ability to eat the magically delicious breakfast of oats and marshmallows or whatever cereal you like. A cup has a similar structure as a bowl, but wouldn't be able to hold both the milk and the cereal. It wouldn't maybe be as efficient as a bowl because it has a slightly different shape. Maybe the cereal would get soggy before you could eat it. And the same is true for proteins. Although their structure may not be familiar as a cup or a bowl, it's important to their function. So each protein's shape basically kind of determines function. And I've kind of preached that since the last set of notes. Shape equals function in biology, especially when we're talking about proteins. So as previously discussed in the course, the DNA sequence is the ultimate source of the structure of proteins. It's basically the instructions on how to make these proteins. It spirals down through the steps of transcription and translation, meaning that the ultimate source of determining the structure of a protein is the order of the nucleotides, those A's, T's, G's, and C's that we discussed. The message that is then transcribed into messenger RNA during transcription goes from there to the ribosome, where it meets up with a tRNA to construct the amino acid sequence like the one pictured on the previous page. This process is called translation. After the polypeptide chain is built, it goes through several modifications to get into its final shape. So what happens if the structure changes? So if we change the structure or shape of the protein, then we're going to change the function. It could cause the protein maybe not to function as properly or as efficiently. So now that the structure's been set, does it stay that way forever? And the simple answer is no. Going back to the bowl of cereal, the bowl can lose its shape if it gets nicked, or if it's a plastic bowl and it gets really warm in the dishwasher, it could melt it or warp it. Pieces could break off, making it less effective, or worse, it could drop and shatter completely, right? And then if it shatters completely, it's not going to function anymore. You can't put milk in it or cereal. So the same can happen with proteins. If it loses its shape, either completely or partially, it's going to lose some of its functionality, either partially or completely as well. Proteins losing their shape is called denaturization, right? They denature or being denatured. So there are several environmental factors that can cause a protein to denature. Temperature is one. pH, the amount of salinity or salt in an environment can change the shape of the protein. If you remember, I think I used an example in one of the last sets of notes. We talked about um, collagen fibers in your hair and how if you curl your hair, if you use a hot iron on your hair, you're changing the shape of the collagen fibers by denaturing them, right? Well, that's temperature. So if we're looking at the temperature of the iron and we put it on our hair and we kind of curl our hair with it, making these loops or curls or whatever we're using, well, that temperature is actually changing the shape of the collagen fibers or the proteins that are found in your hair and making them a different shape, which is why that would work, right? The temperature is causing that to happen. So every protein has an optimal range for how it functions. If the environment changes outside, the optimal range of the protein will lose its shape and then the reaction will slow or stop altogether. So if you take a look at the graph on the right, it shows the enzyme reaction at different temperatures. The optimal temperature for this particular protein would be at its highest point right around 40 degrees Celsius. So if you take a look at zero, absolutely nothing's happening, right? The protein's not working, the enzyme's not working. As temperature increases, the enzyme, basically the rate of the reaction, would increase until you reach an optimal temperature, somewhere right around 40 degrees. And then it quickly, if you start getting up over 40 degrees, you start getting to higher temperatures, it changes the shape of the proteins and the functionality begins to drop again. Okay? So everything kind of has an optimal range. And that goes, if I'm backing up, that's true for temperature, as in the case with the graph. It's true for pH, and it's also true for the amount of salinity that might be in the environment. And there's other things that can denature the protein as well, but those are the three main ones that people usually think of. So if the shape of the protein changes, then the function decreases. Another example is a chair with four legs. It works great, but if you remove one of those legs, then it still functions. The person that's sitting in it just has to concentrate on balancing, right? You can't tilt one side or the other because you would fall. This obviously decreases the function of that chair. If you took all the legs out, well, then the seat in the back of the chair would no longer function, or you'd be sitting on the ground. So if proteins change or denature too much, then they're no longer going to work. They'll either slow down or won't work as well, or they'll completely stop eventually. So what is the impact of DNA on shape? So DNA basically provides the genetic information, which is the original source of all the information in the building of proteins. So what would happen if you changed the DNA sequence? Changing the sequence could cause a cascade of effects all the way through transcription and translation. So if the DNA sequence is changed, if we have a mutation in our DNA and it changes the sequence or the code, then it would also change the sequence or the overall shape of the protein. Changing the DNA sequence is like removing the leg of a chair or chipping away at a cereal bowl. So if you take a look over here, you're going to see, and we're going to talk about it in a little bit, you're going to have normal blood cells, which are kind of like this almost half donut shape with the hole not quite going all the way through. And then you have these sickled red blood cells or sickle cell red blood cells. And there's a disease called sickle cell anemia, and it's a genetic disorder. When you're looking at this, if you look at the DNA sequence, we're talking about one small, slight change in the DNA. So if you take a look, if you have this slight change at one spot on a particular three-letter codon, then it changes one amino acid, and that changed amino acid causes sickle cell. So one little mistake or difference or change in your DNA could cause you to have this sickle cell anemia. So sometimes one change in DNA can cause a big problem, making the protein useless. The picture on the, it's actually on the right, shows how the change even in one DNA nucleotide can cause major problems. highlighted change of the T to an A, right, is all it takes for the body to produce something called sickle cell red blood cells instead of healthy concave disc shapes that I mentioned before. So sickle cell can be fatal because in the absence of oxygen, the cells form a sickle shape, and it makes it so they're not very efficient at carrying oxygen to different parts of the body. And obviously, all the different parts of your body need that oxygen to do things like cellular respiration to carry out their function. So things like your blood vessels can start to clot. It can cause premature heart attacks or seizures. Eventually, even organ failure, and if the organs start to fail, then death as well. So this can be a fatal disease if it's not treated. So how do proteins function? As previously discussed, proteins are responsible for so much in survival and existence of all living things. Remember, DNA is the instructions how to build these, but the proteins themselves are the workhorse, right? In this section, you're going to explore different types of proteins and what they do. You're going to click through, this is on Canvas, by the way. You're going to click through these tabbed info below, and you're going to learn about each different type of protein. So I have just a few here. So on Canvas, when you get to this page, you're going to have a little drop-down menu. So you could click on the structural one, and it would talk about the different structural proteins. And you could click on the transport tab, and it'll have another one, and a motor tab, and so on, and so on, and so on. So there's several tabs that you can click on that you can look at these different types of proteins, okay? So make sure you do that when you're going through the Canvas part. So how does DNA impact function? Again, DNA is the ultimate source of information used to build the protein. It also plays a vital role in their function. Again, this is due to the concept of structure and function. Structure equals function, especially with proteins, and DNA controls what the protein's shape is going to be or structure is going to be. If the DNA is changed, the function of the protein changes as well. On the last page, we looked at an example of a harmful change with sickle cell. However, some changes may be beneficial. For example, if a bug has changed in its DNA sequence for the color of the body to one that might better blend with the environment, it would allow it to survive better against predators that are trying to eat it. Therefore, allowing it to live longer, this means that not only does DNA affect proteins, but it can also affect function. So going back to the sickle cell example, the reason sickle cell is still prevalent, the reason it hasn't kind of went away or evolved out of the population is because actually, in a strange way, sickle cell can actually be a benefit to certain populations. If you are living among people that have, you have malaria is prevalent, right? There's lots of malaria, for example, near the equator or wet areas where there's mosquitoes, because mosquitoes carry malaria. Well, you actually have a little bit of an advantage if you have sickle cell or if you're a carrier for sickle cell, because you become immune to malaria. So that's one of the reasons that you still see sickle cell kind of prevalent. It actually causes, in some strange way, a benefit to certain people that are living in areas with high concentrations of malaria. And we'll talk more about that probably when we start discussing things like evolution and things like that. All right. So what are enzymes? Let's go to enzymes, which are a special type of proteins, right? So many chemical reactions occur spontaneously when the reactants bump into each other or form products. And going back to the hydrogen peroxide, I told you hydrogen peroxide, if you kind of let it set for literally years and years and years and years, it would slowly convert itself or release oxygen in a slow way that would convert it to water eventually, right? But it speeds up if we add an enzyme, if we just put in a catalyst. So often the energy needed to get these reactions started is greater than the living cells can tolerate. So chemical machines or proteins called enzymes make it possible for these reactions to occur. Like a fight promoter brings two prize fighters together but is not directly involved in the fight. The enzyme kind of brings the reactants or molecule together and promotes a chemical change. This could be putting things together or it could be taking things apart like we talked about with hydrogen peroxide. So if you take a look, you have this energy that's required to get basically a reaction started. Now, if you don't have any um, enzymes or catalysts present, this energy might be required to go all the way up here. This dash line might require this much energy to get going. But if we put a catalyst in there, it actually drops the amount of energy we need to get it started all the way down to this solid line. Okay, and we still end up with the products over here at the end, but we don't need nearly as much energy to get it going. So bringing the molecules close together weakens the existing chemical bond and lowers the amount of energy needed to jumpstart the reaction. Reaction will happen faster at a lower energy level than it would if without the help of enzymes. So enzymes allow reactions to occur under normal internal conditions, and then the chemical reactions that we need to function wouldn't necessarily occur without these enzymes because it would occur either way too slow for us to get any use out of it, or it would require way too much energy, which would be a waste of ATP, right? That we would have to use to actually get it going. So if you look at the graph, and we kind of looked at it already, but we'll look at it again. The graph to the right shows how much less energy is required for a reaction. So the dotted line illustrates the energy required for a reaction without enzymes, so it goes all the way up to the top, right? And then the solid line down here is how much energy is required for the reaction with enzymes. Notice both lines start at the same spot, so they're both starting down in here, right? And they finish in the same spot, but the amount of energy required is a big difference between the two. So this illustrates that the products of both the reactions are exactly the same. However, the solid line reaction requires much less energy to run to completion. One of the huge benefits of enzymes is that they can actually be used over and over again, so they can be reused until they are destroyed by surrounding environment or organelles in the cell kind of get rid of them. If they start wearing out or lose functionality, they can be recycled. We take apart the protein and then reuse the amino acids to build other proteins. So they basically get recycled. So how do these enzymes function? So you got to think of there's actually two models that they use. The most common one is called the key and lock model. The lock and key model is basically a generalization on how enzymes work with only one substrate. Just like a key to your house only works on locks in your house, right? And not your neighbor's house. So it's basically if the shape is the correct shape, then the enzyme will do the job. So enzymes kind of also follow another model called an induced fit. And this is probably a little bit better idea. And it's kind of hard to describe, but basically what happens is it kind of, the enzyme kind of changes the shape of the molecule it's reacting on slightly to make it more likely to react with the other chemicals. So unlike the lock and key model, the induced fit model shows that enzymes are rather flexible structures which the active site continually reshapes by its interactions with the substrate till the time the substrate is completely bound to it, which is also the point at which the final form and shape of the enzyme is determined. So it kind of, the enzyme changes its shape to kind of fit. That's why it's called induced fit. It kind of changes its shape to fit the proper substrates, right? And it also helps to change the substrates to fit the enzyme. So what can affect enzyme rate? We already looked at temperature, right? And temperature kind of, as temperature went up, and that would be this middle graph here. As temperature went up, we'd hit this optimal temperature, and then if it got higher than that optimal temperature, then the reaction rate would decrease as well. So that's one thing that can affect reaction rate, right? And we could do the same thing with salinity. As the salt content goes up, it would hit this ultimate range or optimal range, and then it would decrease if we got saltier. Or pH, it hits the optimal range, and then it would decrease if we went higher or lower than the optimal range. So there's several things that can impact reaction rate. The amount of enzymes available. So if you have the amount of enzymes increase, it would essentially keep increasing, right, until we ran out of substrate or out of enzymes. If we got to a point where it ran out of enzymes or substrate, what would happen is it would kind of flatten out and stay steady. The amount of substrate available to interact with the enzyme is also true. So it's kind of the same thing. It would go up indefinitely until you started running out of the substrate, and then it would flatten out. And then temperature, pH, salinity, we mentioned, would be this middle graph here, and that kind of goes up to an optimal range. Typically, the reaction rates are illustrated by using graphs. On the right, there's three different graphs. One illustrates a different type of enzyme reaction. So number one, the one at the far left, the red line here, shows the reaction would continue to increase as we add more enzymes to the environment. This would increase forever unless the environment ran out of or decreased in the amount of substrate available. Okay. The middle graph with the green line shows the optimal range reaction rate, where the reaction slows the further the enzyme is pushed out of its optimal range. This is true for environmental factors like temperature, salinity, and pH, as I mentioned. Different enzymes have different optimal ranges. For example, most enzymes in the human body work best at around 36 degrees Celsius or 98.6, which is normal body temperature. Where enzymes found in deep sea thermal vents, for example, have enzymes that can withstand temperatures of over 400 degrees Celsius. So if you think about when you get sick, you get a temperature, right? One of the reasons you feel really cruddy when you get a temperature, you kind of get body aches and all that stuff is you're basically causing a lot of the proteins that are in your cells that normally are functioning at this optimal 98.6. You're basically, you get a temperature of 103 or something like that, you feel real crappy because you're denaturing the proteins in your cells and things aren't functioning right. So you kind of get, sluggish and achy and all that stuff is because they're pushed out of their optimal range. And incidentally, your body's doing that on purpose. Basically, when you get a virus, okay, the virus isn't necessarily completely alive. It's basically DNA or RNA surrounded by a protein coat. So if we change the pH of our body, we can denature the proteins of the virus and slow its spread too. So it's basically a non-specific defense against infections is why you get these temperatures. The final graph with the orange curve, which shows what happens as we add more substrate, the graph levels off at some point because the enzymes will be working as fast as possible. You'll hit this basically, it's called saturation limit. And this means the amount of enzymes would be called the limited reactant. The same could be true of substrates as well. All right, so that brings us to the final slide and the final lessons or assignments for this week and for the semester. Do all the self-checks again for Module 5, Lesson 3. Do all the check for understanding quizzes. The assignment that you have basically one assignment and then a test, and then we got to study for the semester exam. So the one assignment you have is a 3.6 Protein and Enzyme Science notebook. Make sure you do that. That's where you watch the videos and post a response to the question. And then do the DNA, RNA, protein multiple choice assessment. And also you can start studying for the semester exam. That'll be next week. For all the quizzes and tests for the quarter, I also posted all of the quiz corrections. So if you wanted to work on quiz corrections too to boost your grade, you can do that as well. All right, guys, so that kind of takes us through this week and our final week before we get ready for semester exams. We'll start reviewing for semester exams very soon, probably even maybe this week on Friday. We might work on a little bit of a review or put together some kind of study guide for you guys, all right? All right, guys, you have a good day.